Now that we have created a SR SAM file in the previous video, what basically SAM file is. So sequence alignment map format is a generic format for storing the large nucleotide sequence alignment. So SAM is a format that aims to be a format that is flexible enough to store all the alignment information generated by variant alignment process. You were seeing there were headers there were a lot of things that were in the SAM file that is not originally in our FASTQ file. And so it's a simple enough to be easily generated by the alignment programs and also can be converted into existing alignment formats as well. It's also compact in size. It also most operation on that alignment is work by on stream and without loading the whole alignment into memory. It is also be indexed by genomic position. So SAM is effective to retrieve all the long read and all the short reads aligning to a locus. So what kind of utilities provide these kind of things? So SAM tools are the various utilities for manipulating alignment process. So it's a tool that you can install in your terminal. So for that we are just gonna type SAM tools in my terminal and see yes, SAM tools is installed here. So why we need to use these SAM tools? So how we can install these SAM tools and how we can use these SAM tools. So for using the SAM tools, first of all, we need to have installed the SAM tools. So it was already done in the previous video. So we have created a align.sam file. So let's just visualize that. Get align.sam file. So here is the alignment file that is originally generated here. So closing this. Now we got to change the SAM to BAM file. So what is a BAM file? Basically, SAM file is the human readable format and the BAM file is machine readable format. So we need to convert that. So let's just SAM to and yes, can BAM file is created. So we just use the input of SAM file and created the output of BAM file. So after that we have to sort that BAM file. So we are going to sort BAM file. So let's just run that. So there is a problem with this line. I am just gonna change the directory. Yes, there was a problem. I used the output directory of sort.bam cat sort.bam. Yes, it's a machine readable format file that is created. So now we have a BAM file and we have our FASTQ file. Moving on, how to extract variants from it. Now that we have computed our, we have done the computation and depth coverage. Now it's time to call variants. So how to call variants? So BCF tools. By using the BCF tools, we can manipulate variant call in a variant call format file and its binary counterpart BCF as well. So variant call format is a text file format for storing the marker and genotype data. So every VCF file has three parts that are in order. So first contain the meta information lines and one is a header file and third is the data line contain marker and genotype data. So there is one variant per line. So our data line is also called a VCF record as well. So how to extract variants from these files? Now what we need. So first we are going to fix the nucleotide sites and then we are going to call variants. So we are going to make up a pileup file. It's also a pileup of BAM, your FASTA file, and then we are going to create a pileup file. So let me just take all this code and make some changes here. File.bam, I don't think so. We have the file.bam. Let me just configure it out. So there is sort.bam file. So we are going to use sort.bam name and let me just copy all this and paste it in our text file so now you can configure the code according to our need. file dot fastq, and there is sort dot bam file that is present. So that is it. We need a fastq file, we need a bam file, and then we are going to create the mile. So clear this, all this, and yes, one sample in one input file. So the pileup file was created. So it's a tick sign. Yes, it is saying now it's time to call variants. How to call variants? So we can call variants from the pileup file. So I'm just gonna use this command to create. And yes, note none of the sample files, and assuming all the files were deployed. So let me just read the file if this file has created anything. VCF file dot VCF. Yes, there is information that is presented there. So VCF file contain all the data. So also we are going to remove the homozygous position. So our data should contain some less files. Sorry about that. There wasn't a VCF file. VCF file dot VFC. Yes. So I think there is a problem. VCF file. There isn't any. Yep. It has worked just fine. So it has removed all the heterozygous position and the VCF2 file is created. So moving on, we have to fix the VCF file as well. We have to index the VCF file. Now that we know that how to do all the indexing, and then we have removed the homozygous position as well. We have called the variants as well. So we are going to we are going to index that all the SNPs and indels into our file and into our VCF bundles. So what we need to just run this code. So we are going to index all the SNPs and indels file. So there isn't any file there. So moving on. Yep, VCF2 file doesn't contain that data. So we don't have to do indexing right now because we contain short number of reads and short number of sequences. So that was it for this lecture. In this lecture, we fix the nucleotide side. So we fix the nucleotide side. We call the variants. After that, we have index a VCF file as well. So after indexing, we will be moving on to indels and SNP separation from our file. So that was it for this lecture.